Welcome to our channel by Gulab Sir for Wisdom Coaching Center. As a part of series of lectures, today's topic is implantation and development of embryo. We learn fertilization. We learn that zygote is formed and it is ready now to undergo cleavage and get implanted into uterus for further development of embryo. As it is shown in this diagram, before starting our video, we can visualize this diagram so as to understand some initial stages of development of embryo. We learned that when the ovum, that is secondary oocyte arrested in metaphase, reach to the isthmus, where that second meiosis completed, oocyte is formed and fertilization occur. We learn in detail how fertilization occurs in last video. Formation after fertilization, that zygote is formed, and zygote now after 15 hours of formation of zygote, it undergo division and form two cell stage. Now corona radiata, which was along with that secondary oocyte, get degenerated after fertilization. Here you can see the difference. Corona radiata, fertilization got degenerated. That is the question of NEET. Zona pellucida is continue up to morula stage, and that also question of NEET. So corona radiata till fertilization and zona pellucida till blastocyst formation or blastula formation. So these 2 points you have to remember. Now these are the developmental stages, that 2 cell stage, 4 cell stage, 8 cell stage, 16 cell stage. And this division is a mitotic, but it is known as cleavage. What is cleavage? Cell division without growth. Here the number of cells are increased, but cytoplasm remain same quantity. The quantity of the cytoplasm is the same which was present in zygote. What is increased? That is number of nuclei increased. So nuclear material is increased. Cytoplasmic material remains same because there is no growth. Since there is no growth, this type of mitotic division is known as cleavage, where proportionately the quantity of nuclear material is increased but cytoplasm remains the same. Now here this morula stage is like a mulberry fruit, it's like a solid ball, where this 16 cell. During this cell division, there may not be synchronized division, there may be odd number, but finally that cells get divided and redivide to form this solid ball called morula. And that is what in morula stage that undergo blastulation to form the blastula. Here morula undergo blastulation, that means this each cell of the morula is known as, this each cell of the morula is blastomer. Each cells of the morula is known as blastomer. Now this zona pellucida is about to degenerate or erode. The zona pellucida is about to get degenerate, and here you can see zona pellucida is eroded. That blastomer are arranged in such a way that outer layer of that blastomer form trophoblast. This layer is trophoblast, and here remaining cells from inner cell mass. And this is what we are supposed to learn in process of development of zygote, that is before implantation. So with this diagram, it is very clear to you that zygote undergo division to form 2 cell, 4 cell, 8 cell morula stage with zona pellucida, which supposed to begin to erode, and this morula cells outer layer form the trophoblast and inner form the inner cell mass. Now with this trophoblast, by the time when cleavage was going on, their movement of this zygote in the differential stages was going on. With this movement in blastula stage, by the time it reached to the uterine wall, where the trophoblast cell secretes some content that degenerate some endometrial wall to groove, to produce the groove and get implanted. So trophoblast cells release some material to degenerate endometrial wall cell to form the groove and get in. By the time progesterone also help. That is what progesterone help in implantation. That means all these movement and activities are carried out by progesterone to have the implantation. Now trophoblast cells cytoplasmic processes form the cytoplasmic processes to have these chorionic villi. These chorionic villi are formed all around by trophoblast to get implanted into the uterus. So this chorion formed by the trophoblast along with endometrial wall of the uterine tissue interdigitate so as to have the firm attachment of that blastocyst into the uterus, and that is called implantation. I hope you get it, how this is implanted. This is the very later stage, but initially it got implanted by the process of formation of interdigitation between chorionic villi of fetus and endometrial wall of the mother so as to have firm attachment, that is called implantation. So progesterone prepare entire condition for implantation, therefore progesterone is also known as maternal hormone. I hope you got it. And now these are the different stages of development of this embryo are shown, that also we shall discuss. 2 cell, 4 cell, 8 cell, 16 cell stage, morula stage, blastula, blastocyst, and further there is a blastulation, that was formation of 3 germinal layer from that inner cell mass. That 3 germinal layer are ectoderm, endoderm, and last mesoderm is produced. Formation of 3 germinal layer is known as gastrulation, and yes, we are supposed to understand one by one. Here, to begin, that implantation, as I explained to you, the same I continue to explain here with some diagram. So we learn that this is zygote with this 2n number of chromosome having zona pellucida around it. Corona radiata is degenerated as the fertilization is completed. Now this is ready to divide. Here this is divided into 2 with corona radiata around it. This is corona radiata. Continue. Then 4 cell stage in this way, that lower cell has is divided into 2 cells, again 4 cells. These are the 4 cells, one is equatorial and another is peritoneal. So this cell division, again this is 4 cell stage. Now 8 cell, so it can be shown, either shown by this as an explanation given in references, or you can show as it is, that what mentioned in your book to avoid the confusion. Here 4 cell stage, with zona pellucida. Again 8 cell stage, cytoplasm content is same, 16 cell stage, remaining cells are from that side, so need not to have that exact count in the given diagram, but it is very important to understand the different stages. See, zona pellucida is continue even in morula stage. Zona pellucida. Now this each cell has a diploid number of chromosomes. So that nuclei consist of diploid number of chromosomes. As that cell undergo mitosis, where this is what cell division by mitosis. But because of without growth, this is also called as cleavage. There is no increase in the content of the cytoplasm, only nuclear material is being increased. So this is called as cleavage. So finally, what the morula is produced is gives mulberry-like structure or solid ball having all the cells. Now each cell is known as blastomer, and the stage is morula. Now why this is called blastomer? Because each cell later form the blastocyst. What is that blastocyst? Having arrangement of this outer blastomer arranged to form the trophoblast. So to have this, so these cells which arrange in outer zone forming a layer called trophoblast, and this inner cell mass, that what can be shown by this is what inner cell mass, also called formative cells. So can be labeled as inner cell mass. This is a trophoblast outer layer, and this is inner cell mass, also called as formative cells. Formative cells, because they form the base of the embryo, 3 germinal layer. Now by the time that zona pellucida is degenerated, so here we can show that the zona pellucida is being degenerated by the time. So zona pellucida is erosion of zona pellucida. Now this is very important to understand how long corona radiata is continued to be with that secondary oocyte till fertilization, and how long zona pellucida is continue up to blastocyst. Now this stage is called blastocyst. This stage is called blastocyst. And here this cavity which produce is called blastocele. And this inner cell mass cell now arrange and rearrange. Now this process of conversion of morula into blastula is known as blastulation. So this stage of conversion of morula into blastula is called as blastulation. Now after formation of blastula, blastocyst, it undergo gastrulation. Now what is this gastrulation? Process of formation of 3 germinal layer from this inner cell mass. By the time this trophoblast form chorionic villi, and that we can show like this. Now to understand again, see, these are the finger like projection developed by each cell of this trophoblast. These finger like projections are chorionic villi developed by these cells of the trophoblast. These finger like projections are chorionic villi that help to how the implantation. So interdigitation of this chorionic villi with maternal tissue. Suppose maternal tissue we shall show by this way. Here we can show maternal tissue by this way. This is a maternal tissue of endometrial wall. So interdigitation of this endometrial wall of the mother make that a form implantation. So this we just understand that this is inside the uterus. This is happening inside the uterus. So this implantation, that interdigitation, this endometrial wall of the mother is provided blood vessels so as to have the supply of nutrition by diffusion to the developing this chorionic villi cell. At the same time, what happening inside this inner cell mass, that very first layer of inner cell mass form a amnion, that cells present towards upper part, outer part form this amnion with amniotic fluid to protect the developing embryo within. That some of the cells of this inner cell mass form the ectoderm. So this outer layer is called ectoderm. Now this ectoderm form some organs like nervous system, skin, and all these 3 derm together form whole body. So ectoderm. Now first that ectoderm is produced. Second, this endoderm is produced. This is endoderm. And third, last, this mesoderm is produced. Mesoderm is with mesodermal cavity, that is coelom. So here this is a mesoderm. With cavity, having cavity in it. This is a cavity called coelom, where the organ system can be arranged. Now formation of 3 germinal layer. This stage is called gastrula. And the process is known as gastrulation. Now here these three germinal layer are continued to form entire organ system. Now here the embryo is almost implanted. Trophoblast, amnion, ectoderm, endoderm, mesoderm. The sequence of formation of layer is very important. First the ectoderm is formed, then endoderm form, and later mesoderm form with cavity coelom. Therefore our embryo is triploblastic coelomate that we learned earlier. Now that some of the cells of this ectoderm, they continue to divide and redivide to form the placenta with the interdigitation with the endometrial wall. Now placenta is provided with umbilical cord, that is blood vessels, with very close to the blood vessels of the mother. So capillaries of the mother get interdigitated with very close vicinity to the capillaries of the fetus. And now fetal arteries and vein, that is umbilical cord, help to have exchange of gases and transport of gases as well as nutrition and waste material, that is shown in this diagram. So I hope you will understand very well. See, this is a umbilical cord, see, this is a placenta, and now there is a interdigitation of, see, mother blood vessels and these are fetal blood vessels. There is a exchange of, that is what oxygen is being transferred. Oxygen is being transferred from mother body to the fetus, and carbon dioxide is transferred from fetus to the mother body. Nutrients are transferred from mother body to the fetus, and waste material. So waste material from mother body is removed. So here you can understand, nutrients are given to the fetus, waste is given out from the fetus, oxygen is given to the fetus, and CO2 given out from the fetus. So exchange of this content occur in between that blood vessels of fetus and blood vessels of mother through interdigitation of chorionic villi and maternal chorion and fetal chorion. I hope you did understand how this implantation, after implantation, that umbilical cord and placenta is developed from that ectodermal tissue. Ectodermal tissue means which one? Here. So ectoderm that form the placenta. So placenta is associated with umbilical cord, where that blood capillaries of fetus get in interdigitation through chorionic villi. So maternal and fetal chorion interdigitation with these blood vessels of mother and fetus come in contact for the exchange of oxygen and carbon dioxide and waste material and nutrients. Now implantation is completed, and now development after that implantation within 1 month. For the first month, if we write there that changes which have occurred, that what in first month heart development is completed. Here that fetus with first month, that 5 week is given, by the time that heartbeat can be noticed. So here I do write that heartbeat can be noticed in first month of development. Heart development, that is circulatory system is completed and heartbeat can be noticed. So this is what first. Now second month, development of limbs. See here, development of limbs by the time of 10 week to 20 weeks. That means 10 weeks, it almost 1.5, 20 weeks, that is around. So here by the 10 weeks, that digits, what we call second month, by the complete of second month, that digits or limbs, development of limbs with digit. Now after 12 week, that is first trimester, in between this 20 to 10, after 12 week, I write here 12 weeks to understand that most of the major organs are developed. External genitalia appear. So here I write external genital organs appear, and limbs are completely developed. See, legs and hands are completely developed. That is after 12 week. That is my first trimester. This is completed. Now 5th month, then by the time of 5th month, what is given in your book, according to that, by the time of 5th one, first movement of the baby. So here first movement of the baby is appear. First movement of the baby is noticed when of the 5th month. and also that hairs appear on the head. Hairs can be shown here. Hairs begin to appear on the head. So hairs appear on the head by the sign of 5th month. So movement of the baby, first movement of the baby, and hairs on the head are appear by 5th month. And by the second trimester, that means 3 months are completed. So by the second trimester, we can write here, by the second trimester, it is not actually, this is fully developed, but by the second trimester, eyelid, eyelashes. So to know that, I can write here, by the time of second trimester, that means after 6 months, so there that eyelids separate, eyelashes appear, and hairs on the body appear. And by the end of 9 months, fetus take the baby. Once again, to repeat the developmental stages, first month heartbeat is being noticed. By the second month, that limbs and digits are developed. By the end of the 5th month, first movement of the baby, and hairs on the head are appear. And by the end of second trimester, that what eyelid separate, eyelashes appear, and hairs on the body begin to appear. And by the 9th month, fetus get completely developed to have the birth. So I hope that you did understand the process of implantation and development of baby days and month wise. So you can refer it to have the complete idea about the development of baby in first month, in second month, in say 5th month or second trimester, and finally 9 months when completed, that is called gestation period, is completed and baby is ready to take the birth, and that process is known as parturition, that we will learn in second video. I hope you did understand the process of implantation, that is embryonic development and implantation, that also you learn about that how fetus is developed in that 9th month and get ready to take the birth. Even if you are in doubt, that you can ask in comment section. I appeal you to like our channel as well as subscribe it. Also press bell icon to receive new videos as and when they are uploaded. Thank you.